So it's a pleasure to be here. And I am not an expert in regulation. I'm a molecular biologist and microbiologist by training. And somehow I represent the academia because I lead the biotechnology and synthetic biology research group in the University of Valencia, based in Valencia, Spain. And I'm also the co-founder and CEO of the company Darwin Bioprospecting Excellence. We basically develop microbial solutions for the industry. But I think I cannot hide anymore because if you type my family name on the internet, you will very fast realize that I'm the guy that was awarded an Ig Nobel Prize for describing the microbial communities of the wasted chewing gum. So it was me, yes. So going back to reality, one of the things that I've been working for a while is standardization in synthetic biology. So I coordinated a EU-funded project called BioRobust, the goal of which was fostering standardization in synthetic biology. And I only have 2 slides because I have just 2 main messages that I would like to share with you today. The first one is this one here. So what can be standardized in synthetic biology? The short answer is everything. And as you can see here, the first thing we, molecular biologists and microbiologists, think when we talk about standardization is very likely DNA parts, so genes, promoters, etc., that can, of course, be standardized. This can be combined into devices that at the same time can be combined into circuits. Circuits can be combined into pathways, but these pathways are not floating in the air. Finally, they have to be introduced into a living cell, typically a bacterium or whatever. So you have one engineered cell. Then, of course, you can work with several cells in a consortium. And all those things are subjected to protocols that can themselves be standardized as well. And finally, of course, there are human practices. So there are people that pipetting in a different way sometimes, and you know that there are standardization efforts that have placed TV cameras in the labs to see what are the different physical maneuvers behind the success of some technicians compared to others, for example. And one interesting thing is that not only there are several levels, almost infinite layers of complexity that can be standardized in synthetic biology, but that the structure of such levels is not unlike the Russian Matryoshka dolls in such a way that every standardization level is included into the next one. So the other point I want to stress with you is where are we in terms of standardization in synthetic biology? So this is a scientific paper that we published in EMBO Reports like a couple of 3 years ago. And this is one of the flagship reports of our project BioRobust. That is a European Union funded project, as I mentioned, but there were also partners from here, from the States, from Asia as well, from Singapore and from Japan, 25 partners in total. And one of the, I think, more visually appealing conclusion that we wanted to describe in this paper is where are we in terms of standardization in synthetic biology? The short answer is not yet very far away, but progressing nicely. So just to describe 3 of the entities, let's say, that are plotted in this figure. First, BioBricks. BioBricks stands a bit left in this graph because it is true that it's, as we all know, not widely used anymore, but BioBricks will always have the merit of being historically super important. They were the basis of synthetic biology. They were linked to iGEM, and conceptually and historically they will always be there. At the other, almost at the other extreme of the graph, there is the SIBA plasmids that has been designed by a colleague of mine, Victor De Lorenzo in Madrid, that are plasmid. SIVA stands for Standard European Vector Architecture. So good thing about SIBA is that even there is not a strong regulatory effort behind them, they have become like de facto standards because they work very well and they are shared at no cost, by the way, by them and used in many laboratories and hundreds of laboratories, I would say, worldwide. And of course, SBOL, the synthetic biology open language, which is widely used, I would say, right now. And additionally, it's shared between this discipline, synthetic biology, and its sister discipline, systems biology. Good news is that this article, this plot was made like 2 or 3 years ago, but right now I would say that most of those parameters would be a little bit more towards the right, so the progression is quite nice. So that's what I wanted to say. I'm super delighted to be here, and I hope that you found this contribution moderately interesting, considering the amount of kerosene that was burned to bring me from the other side of the ocean. So thank you.